Good evening and welcome to the NPTEL course of Proteins and Gel-Based Proteomics. This specific course has a code of BT57 and this is week 4 lecture. We have already done with 3 weeks of lectures. So before, so in week 4 we'll be basically dealing with differential gel electrophoresis. That is specifically Dice technique, and we'll also be dealing with mass spec a bit, mass spectrometry technique. So before going into details of week 4, let's just recall what all we did in week 1 to week 3. Can anybody just brush up like what were the topics we covered in terms of week 1 to week 3? Yes. So we majorly dealt with initially we dealt with what amino acids are, then what proteins are, uh, the structure of proteins, etc. Bit of introduction of proteomics, and then we went ahead to talk about sample preparation, how exactly sample needs to be prepared before performing any type of proteomic analysis. And then we went ahead to do gel-based proteomics technique, one dimensional and two dimensional. So in two dimensional, there is isoelectric focusing, and then there is the age that is agarose gel electrophoresis. And now in this week 4, we'll be dealing with another gel-based proteomics technique, which is DIGE, which is the name itself tells you differential gel electrophoresis. So we'll basically dealing with differential expression in different samples. That all those things can be done in one gel itself instead of doing using multiple gels. Like if you use this sample analysis by two-dimensional gel electrophoresis, you will be able to see like you need to run multiple gels accordingly depending upon the type of samples. Whereas if you are using dyes, the benefit of using dyes is like you can pull up multiple samples together and run one gel itself. And within a gel, you can visualize it differently what might be present in each sample. That's what DICE is technique is. So in this week 4 lecture, we'll be majorly dealing with DICE technique only and a bit of mass spec. There will be in so there were 5 lectures which were uploaded for the same, wherein the first part was about basics of what DICE technique is all about. Then how can we analyze data using DICE? Like if we have done DICE uh, technique, then how do we analyze the data after obtaining the results? And a bit of case studies and applications of what diets can be used. So it will have similar application as that of Proteo, like the case studies which we have done for two-dimensional electrophoresis, but in a different way. Similarly, after that, a bit of protein identification. So dyes will only tell you that specific molecular weight protein is present or not. So that same specific molecular weight of protein might be different protein also. Like we never know, like having similar molecular weight, but there might be different protein also, or else which what is that exact protein of that same specific molecular weight. So in order to identify what exactly that protein is, in order to prove that that's what your protein of interest is actually present in your sample and it's there. So you need to use mass spec analysis. We'll be dealing with protein identification using MALDI-TOF, which is a mass spec technique, and a bit of proteomics experiment data analysis and challenges associated with that. So those were the 5 parts which Sir talked about in this week 4 lectures. So what is DIGE? So I just wanted to confirm again, like if my slides are visible properly. I think someone messaged as well. Okay, I think it's done. It's there. Fine. Where is the slide? Okay. So, what diet is? So it's basically, I have already given you an overview, but what diet, who established this method, etc. So, it basically to overcome intrinsic gel to gel variability of two-dimensional gel electrophoresis, as I mentioned, like if you are doing 2DE, then per sample you need to run one gel itself. So gel, there will be a possibility that one sample was run a little greater, or one sample analysis was not that was not done similar to that of the first one. So gel to gel variability can occur. So in order to avoid that, DICE is a multiplexing technology merged together. So all those samples can be pulled together and run at one go itself, and that's what the major benefit of DICE is. So this method was initially reported by Unlu in 1997. You can go through this specific paper as well. A protein labeling and separation technique. That's what this technique is. So you basically do labeling first and then separate it. So this is the step which is additional in terms of doing dyes. Then detection of protein with fluorescent tags. So this labeling are fluorescently labeled tags, and you will accordingly determine those specific proteins with the fluorescence it is giving. So how exactly fluorescence is detected, you all might be knowing already. Like you need certain excitation wavelength, and and it will emit certain wavelength which can be detected. So in a similar fashion, such proteins can be detected, and it is a linear detection in wide range of protein abundance. So what are the commonly used fluorescent dyes? These are Psi2, Psi3, and Psi5. It is nothing but hen-hydroxysuccinyl ester derivatives. So 2, 3, and 5 are those derivatives of cyanide dyes itself. And these are the commonly used dyes in terms of this specific technique. And so they label samples by using this technique, by using this dye specifically, and they can co-migrate within the gel itself. Like that, those dyes can also migrate with your sample. So these are the three specific dyes: Psi3, Psi5, and Psi2. And the benefit of these specific dyes is like they are having a relatable molecular weight. So as you're doing separation, because of molecular weight, it will not give any of much difference between that molecular weight, because it is having similar molecular weight if you see. So that's what helps in terms of this specific using these specific dyes. And another benefit is each and every dye has its own excitation and emission wavelength. So, accordingly, the sample which was exposed with this specific dye, you can observe those samples by giving these excitation and emission similarly the other ones. And along with that, spectral distinct, this indicates the same thing, spectrally distinct, indicating they are spectrally different. If you can see here, they have excitation and emission wavelength different, and they're also pH insensitive. That's the reason you can use isoelectric focusing as well. So it will not be because of the pH change in pH, it will not be getting affected while doing isoelectric focusing, and it is also photostable. The reason of using Psi2, Psi3, and Psi5 is because it does minimal labeling. But basically, labeling technique can be two different ways. You can either do minimal labeling or saturation labeling. Majority of the time people do minimal labeling only, so that it doesn't affect the protein much, and it will not affect the protein structure and all the stuff. So generally minimal labeling is done, but sometimes depending upon the aim and need, people can go for saturation labeling as well. And in minimal labeling, the amino acid which gets labeled because of these dyes is lysine, and because they have charge like similar molecular weight, the size is matched and no multiple labels are done with the help of this technique. Whereas in terms of saturation labeling, there are multiple labels, like different amino acids are labeled, and that's what how cysteine labeling happens in terms of saturation labeling. And this is the more sensitive technique, type of labeling technique as compared to that of minimal labeling. So why exactly lysine is preferred? Because if you see, if, as we need minimal labeling, and like for example, you might have a question like why if you want to label the protein itself. So this basically chart indicates that generally in a protein, what type of amino acids are generally present. So generally for an amino acid, tryptophan are very less for majority of the proteins, but on an average, lysine was selected for is a preferred amino acid to label because it is generally present in more than 5%. On an average, it is generally present in most of the proteins. because it is almost present in every protein as it, and at least have one lysine residue in the whole protein. And in minimal labeling, you are generally labeling 2-3% of it only, because it will not affect the entire structure of the protein, etc. That's the reason lysine is selected. Then you might have a question like, why not leucine? Because it is, as it is present 10%, as it is present in 10% of the, like more than 10% of the those proteins, etc. So why don't we use this? Because it might label majority of the protein, and it can affect the structure of the protein, etc. So all those can come into picture if you are doing extensive labeling. So this lysine was chosen as the preferred one because of these benefits. So generally, these cyanide dyes react with primary amine groups. And another benefit of lysine is basically it's a polar amino acid, and it is generally present outside the, like it's generally present on the surface. So even dyes can easily interact with lysine compared to that of hydrophobic amino acids. So even that's one of the reason. So it reacts with primary amine groups and terminals. So we'll go through the reaction in this next slide. This is how the interaction happens. So with the primary amine group and N-terminal alpha amino and lysine epsilon amino groups, and basically nucleophilic substitution is done while this interaction is taking place. And it's generally all available lysine labeling would create a very hydrophobic protein. So minimal labeling is generally preferred, because if you label everything, then it might create a very hydrophobic protein, and because of that, all the separation techniques which you will be using that can get affected. So minimal labeling is done, wherein only 3% of the whole proteome is labeled, because so this is what helps. Like that's the reason lysine labeling is done. How exactly this labeling happens? So your protein has this amino group outside of that specific lysine amino acid, and that basically interacts with dye and NHS ester group. So it basically this side dye. This is how the dye looks like. This side dye fluor, that's the fluorescence basically. It is positively charged, and it has a linker. This specific part is called as linker, and there is NHS ester reactive group. That is what is reacting with your protein. That amino group is reacting this specific group. So, and at pH 8.5, NHS ester couples with the amino group of lysine. So you need this specific condition in order to react this specific dye, and it forms a covalent amide bond. So by giving this condition, that dye and your protein of interest gets interacted, and that's how it gets labeled. And the amine group of lysine residue is positively charged at neutral or acidic pH. Just a minute. And so generally this dye is positively charged, so the net charge on the PI is effectively unchanged, and we were also discussing that these are not exactly pH unstable, or what was that point? So it is pH insensitive, so it doesn't get affected by pH as such. And these two are separately resolvable dyes for mass to charge ratio. So generally these two are used for labeling the samples, and the side 2 is generally used for labeling the control. So you generally, for in terms of sample preparation, these dyes are uh, dissolved in DMF first, and then they are used for labeling a protein. And we have already done with like how exactly this buffers are used, and this much amount of protein can be labeled using this specific dyes. Just a minute, people. Yeah. So around 50 microgram of protein is labeled. That much amount of protein can be labeled by using these dyes, and by, as soon as you add the dye, you need to quench this with excess of primary amine. So that free lysine, 1 microliter. So this is the basic concentration is required in order to label your sample. So this whole is involved in terms of sample preparation. Then Generally, having an internal standard is important while doing any type of proteomics or mass spec analysis. So, what does internal standard is all about? So, in terms of mass spec, internal standard can be any specific compound, for example, which will test whether your uh, method or sample, whether your method of LCMS or GCMS mass spectrometry is working fine or not, because that internal standard, so basically it's a calibration step kind of a stuff, to check whether your instrument is running smoothly or not, because that specific internal standard should give you same results every time as whenever you are running your samples. Or else internal standard can be other way around also, by pooling your all the samples. So for example, you had certain control sample, you have treated sample, you will pool each and everything in one sample itself, and you can, so basically this pooled sample is also called as QC, like the quality control, or else it can be also called as internal standard. So this, so in this specific internal standard thing, whatever metabolites, whatever peptides you will find in terms of proteomics, in terms of metabolomics, it's metabolites. So in terms of proteomics, whatever peptides you will find in this pool, only that will be considered in your samples. For example, if sample shows some unknown peak or something which is not present in the pool, you should not consider that because it was not present in your internal standard itself. So that's basically internal standard helps you all to dismiss the unwanted peaks or unwanted peptides you might end up getting in your samples or in your control, which is not present in your pooled sample or a quality control or internal standard sample. So this will act as a control, and it will tell you, give an comparison that if there are any contamination happening afterwards while preparing the sample, etc. So that can be neglected, and if they are visible in your spectra, those can be cancelled out. So having an advantage, that every protein in a population appears on each gel. So having an internal standard, so you can see all the protein in each gel itself, and gel to gel variation will get reduced. Gel to gel match and comparison becomes much more easier because it should present in your internal standard gel, no matter what. And if it is not there, you need to eliminate that. Similarly, a reference point for every protein species will be present on your QC1 because it's pooled and it will have everything in it, and it generally helps in automate accurate spot statistics can be performed if you have internal standards. So in the software itself, you can tell that consider only these proteins in all the samples whichever been tested. So accordingly, that helps in a later stage. So how exactly these are prepared? Like your sample one, sample two, and internal standard. So so sample one can be control, sample two can be the diseased one. Depends upon your study. So as I said, like Psi 3 and Psi 5 are generally used for your samples, and Psi 2 is used for labeling internal standard. You can pull all of them together, and then this sample can be used for running 2DE, which is so basically this labeling step is the additional step, and all other steps which we have already discussed in 2DE. So your sample preparation changes in terms of you are doing differential gel electrophoresis, wherein you will be using specific dyes for specific samples and you will be pooling them together. So this internal standard is already pooled. And while preparing the dyed sample, you will again pool it with your samples as well. So when you are visualizing Psi 2, like be giving certain emission and excitation wavelength, you will visualize this. So this will have each and every protein, and you need to compare this specific gel with your these samples and only consider those proteins which are present in this, and not if they are exclusively present in one of the sample, you need to eliminate that because they are not present in the internal standard. So you'll run the isoelectric focusing. You will first rehydrate the gel, make it activated, and then you will run to the isoelectric focusing. Depending upon the PI, it will be getting run. So now dyes will not be getting affected as we just discussed, and you will equilibrate it with reduce it and alkylate it. After that, you will be running the SDS page, and that's how proteins will get separated. Now, in terms of visualizing this, so now it changes. So in generally after this, you will, in terms of 2D, you will either stain it with Comasi blue or silver stain or Procure diamond, etc., depending upon your need. But now, after that, you need to. So this was the first part of the lecture, wherein Sir talked about different dyes, how exactly this dye is performed. So now, while visualizing the gel, this specific excitation and emission wavelengths are given, and the gel is the colored which is observed is these specific colors depending upon the dye. So these are just the second part of the lecture wherein Sir discussed about the analysis technique. So how do we analyze it? So now you have run the gel. Now you are visualizing and analyzing it. So by for visualization, you will use similar excitation, and so it is already labeled. So you don't have to dye it and etc. Either chromatic blue staining or uh, silver staining, etc. You don't have to do that. So now you will directly visualize it depending upon the excitation and emission wavelength, which we just studied. So internal standard is labeled with Psi2. Three images can be obtained by three different excitation emissions, Psi3, Psi5, and Psi2, by using the same scanner which we studied. Now the decider software used for image analysis. So this is the software which can be used for doing image analysis. Now this will, there are other softwares as well which minimizes gel to gel variation. This BVA indicating minimizing biological variations. This can be further analyzed using statistical tools. So there are different tools and in terms of analysis which are available. You can use these tools and this specific software for doing specific type of analysis depending upon your need. So this is just the labeling protocol, your cell surface protein labeling in intact cells. So you can do it. This specific type of labeling can be done on intact cells as well. And for cell surface protein, it can be done for cellular proteins as well. So now you will lyse the cell, which we generally have learned about the technique, right? Like you will take the sample, you will lyse it, like while sample preparation. But this specific protocol can also be used for intact cells as well. So these were the specific dyes you will be used. There is an incubation process. Then whichever are unreacted, you can quench it with 10 millimolar lysine so that whatever is the dye which hasn't been interacted with your protein, that will interact with this lysine and it will stop reacting with the protein more. Then you will do two-dimensional gel electrophoresis accordingly and you will do the analysis. So how do we do analysis? So those you will take those gel images after visualizing them like at different excitation emission wavelengths, Psi2, Psi5, and Psi3. Now they are uploaded in the software, the decider software, and then the changes will be made to Psi2 images are extrapolated to Psi3 and Psi5. So what does this mean? So Psi2 is your internal standard. Then that specific gel will be used for checking whether these two specific sample dyes having similar spots or similar gel separated proteins, and if there is any difference which is not present in Psi2 but present in Psi3 and Psi5, they are eliminated. Then that's how this analysis can be done. So those were the short forms basically. So DIA is used for minimizing gel to gel variation. BVA is for minimizing biological variation. So one set of all these three images will be taken. 3D view will be taken of all of these three images, and slope and etc. will be calculated. So that was a part of in your second part of the lecture. Then in the third part, Sir digged in more about how exactly analysis can be done. So now we have run the samples. There is an internal standard. There are sample one, and so this is the internal standard here. Green one, Psi2. Psi3 is your maybe control sample. Psi5 can be your disease sample. They are mixed together. Isoelectric focusing is done. Then 2D like SGS page is done. Then now you're visualizing depending upon excitation emission. This is how the proteins were seen, for example. So if you see, so this is your internal standard. So this protein is specifically coming from the control sample, but it was not present in the disease sample. So if you want to identify this protein and check why it was not present in disease sample, so you'll understand like this is a protein which is generally responsible for keeping the control sample safe, and in disease sample, for example, basically those specific protein is not at all produced, and that's the reason that person might be suffering from that disease, etc. Similarly, if you see this specific protein is also present in control only and not in the diseased one. So that's how you can compare these results and make sure that it is already present in the internal standard as well, in order to make sure it is not coming from anywhere, but it is coming from the sample itself. And that's how you can do the analysis, and just by visualizing. But we can actually know the intensity, etc., by uploading it into the software and check everything about it. So this is just the whole protocol, what can be done, like collecting the blood samples, then separating the serums, etc., and then doing the analysis of 2D gel electrophoresis. So in 2D, you will have to run multiple gels, like run in order to analyze all patient samples. Several 2D gels will be required. But in terms of DIGE, you can, for example, these many were control samples, these many were test samples. You can also pull all of these, and the separation can be pulled together, which acts as an internal samples. Is anyone asking anything? Or just okay, all good, right? Till now? Yes? You can show thumbs up, etc. Write it in the chat box. Yes, people? Should I go ahead? Yeah, thank you. So what happens here? So you are it's just the revision of the same thing. Then you will visualizing them using different excitation emission wavelength. Then this is how it will be visualized. This is again an overview basically. So you are basically mixing, doing isoelectric focusing, and then 2DE, and then doing the analysis. So this is how the gel looked. Then by when you viewed it inside the decider software, by giving certain excitation and emission wavelength, then you can only visualize those proteins. Although in this specific sample, as it is pooled one, it will show like the proteins, all the proteins basically inside this. But while visualizing that specific protein, you will only see. So these are the same gel basically. You are running only one gel, but you are visualizing it differently. So in that gel, basically all these proteins will be present together. I hope you all are understand. If not, then you can ask me the question. So we are running only one gel itself by using all of these sample. Like basically, you are pooling it together. So, but what was present in sample one can be decided, like can be visualized by giving certain excitation and emission. Similarly, what was present in this sample two, different excitation emission can be given and visualized. So you are running only one gel, so multiple gels are not required if you are using dyes. And then you can directly visualize them in that one gel itself, your internal standard, your control sample, and your disease sample all together at once. So gel to gel variation is minimized because in 2DE, you need to run control differently, you need to run internal standard differently, you need to run uh, your disease sample differently. The three gels will be required, for example. But here, one gel and all of those can be visualized separately by just giving excitation and emission differently. It will tell you like positive control has that specific. So we already done this analysis similarly, but in terms of dyes also, you will able to get each and every protein expression separately, like whether it is highly expressed or like down regulated or up regulated, etc. All those can be analyzed using that specific software. So it's an extremely valuable technique. It can analyze large number of samples. It can overcome the problem of gel to gel variation, etc. So now Sir discussed a bit of case studies as well, wherein Sir talked about how exactly this dice technique can be used for doing analysis. Okay, so this is one specific study wherein Sir talked about prostate cancer patients. Mhm. That is Deitch technique. Like they have used this specific technique in order to identify any specific markers in prostate cancer patients. So they have used healthy control and prostate cancer individuals. So generally, this is PSA is prostate specific antigen. Generally, that specific antigen is determined and checked whether a person might be suffering from prostate cancer or not. There is certain levels basically. If that much amount of prostate specific antigen is been seen, then people say that that person individual might be suffering from prostate cancer. Then further cancer specific analysis are done in order to make sure that that person might be suffering. But that's the general biomarker which is used. But the problem with PSA is it can also be present in healthy individuals as well. For example, he is due to some or the other reason, sometimes even a normal individual can show higher amount of PSA. So that necessarily doesn't indicate that the person is suffering from prostate cancer for sure. So this study wanted to find any other specific markers which is specifically related to prostate cancer only. Like if a person is suffering, then only that uh, protein is higher in amount, and or else it is not. So they wanted to determine or find a few other biomarkers. So they collected serum. We already know that in serum, albumin and globulin is generally higher in concentration. So those are separated first, high molecular weight ones, and then so that even a smaller concentrated sample can be detected by the proteomic analysis. So in this specific analysis, they have used disease sample itself. So they have separated that disease sample into two, cohort 1 and cohort 2. Why they have separated this disease sample itself? Depending upon the severity of prostate cancer. So that was determined by giving Gleason score. So Gleason score is basically used in terms of determining prostate cancer individuals, wherein depending upon the stage, if we whether it is an early stage or later stage, depending upon the stage, a specific Gleason score is given in terms of that specific cancer. So they have divided this disease sample itself into two, like one which is severe and one which is less severe, depending upon the score. Like basically stage, I mean to say, one which is might be at a later stage and one is at a early stage, for example. So those two samples were taken, they were pulled together, isoelectric focusing was done, SGS page was done. Now we need to visualize those samples. So when they so here they have done 2D electrophoresis only. So now this technique we are talking about not dyes exactly, but we are talking about 2D electrophoresis now. But here the aim is to identify which protein it is. So now they have done 2D electrophoresis by running two different gels, and when they have run these two different gels, they try to determine which might be. So these are certain proteins. If you see, they are present here, but they are not present here. So accordingly, they took those specific proteins and done a mass spec analysis for identifying those specific proteins. So how exactly mass spec is done? So when they have, we'll deal with that in the next part of this lecture. How exactly mass spec analysis is performed. But when they did those specific analysis and they found that these were the few proteins like pigment epithelial derived factor, uh, ficolin 3, etc., polypoprotein, etc. So few were down regulated and few were up regulated in this differential protein expression between these two by using decider software. But now they know that these were down regulated, these were up regulated, but how much it was down regulated or how much it was up regulated, etc., we don't know that. So basically this is a research paper which is being shown here. So they have done further analysis for these two proteins, like pigment epithelial derived factor and zinc alpha glycoprotein. So these were the two proteins. If you see, like these were down regulated and these were up regulated, right? So now they wanted to see how much it was up regulated or down regulated. So they found that in these two groups, like the cohort which they tried to see, so in this Gleason score 5 individuals, it was higher as compared to that of the other one. So they have used ELISA technique, wherein that specific uh, anti-protein of that specific protein is present, and depending upon the concentration, that much will interact with this ELISA. I hope you all know how ELISA works. So that much amount got interacted, and accordingly concentration was determined. Similarly, they also did zinc alpha glycoprotein, and they found that in these two groups there is no much of a difference, but it is slightly upregulated, but not much different in terms of getting upregulated in the other cohort. But whereas in terms of this, it was the Gleason score 7 individuals was completely down regulated. We can clearly see that compared to that of Gleason score 5. There is another technique which is called as immunohistochemistry, wherein a tissue sample is used, histoindicating tissue, and if the tissue is expressing any specific type of protein, then when you are adding the serum, that will have that specific anti-protein to it, and when you will do the analysis, you accordingly the secondary antibody which is tagged with certain substrate, basically tagged with certain enzyme. When you add a substrate like chromogenic substrate or chemiluminescent substrate, accordingly you can visualize if your protein of interest is present in the tissue sample, then accordingly these interactions will take place and you can see the uh, protein, like visualize the protein. Or else if there is no protein in the on the tissue, then you will won't be able to see that interaction and it cannot be visualized. So they found these two proteins as the markers, PEDF and ZAG. Like those can act as markers in terms of prostate cancer. That's what they found in this study, and it can be a potential biomarker for early stage prostate cancer prediction. That's what they found, and that was about one of the application using this specific techniques. Now we want to identify the proteins, although they have done this, but how exactly this mass spec is performed. So, in terms of mass spec, also there are different ionization techniques available. In that, we'll be dealing with matrix assisted laser desorption or ionization technique, which is MALDI. And in terms of detector, like after ionization, how we'll be detecting those ions. So there are different ways of detecting as well. So in that different ways, we'll be majorly dealing with time of flight only, like it's a type of detector. Not no, we cannot call it as detector maybe, but it is a type of analyzer. It analyzes the ions, and depending upon those analysis, we actually that's what it will hit the detector, and then we can analyze uh, what type of protein it might be. Then in this lecture, sir also talked about a bit of how exactly the samples are prepared. Like in gel digestion, we already talked about this in the last lecture too, when one of the individual asked out how exactly mass spec works. So now this will be the third part of the lecture, but I hope everything is going fine till now. Yes, people. Yes, Neha, Veena, Himani, and Anish. Going good, right? Till now. Yeah. Okay. So what mass spec is all about? It basically we are separating the peptides by fragmentation, and it's the separation happens depending upon mass to charge ratio. It is a technique for production of charged molecular species. So after ionizing, you're giving them certain charge. So depending upon the mass of the peptide, etc., the charge will differ, and accordingly, because of that, mass to charge ratio is done, and then the separation is being taken place in terms of this technique. The separation by magnetic and electric fields. That's what happens in the top basically. So what do we do in this? We ionize it, separate on the basis of mass to charge ratio, and detect it depending upon the spectrum which is observed. So generally a proteomics experiment involves 2D or dyes and then identifying that specific protein. So when after doing this 2D or dyes, you will analyze the results, and you'll find that one of the protein here was upregulated compared to other, or one of the protein here was downregulated compared to other, etc. So what you will do, you will take that specific band of the on XGS page, and then take that into in gel digestion. Like after analyzing the gels, one needs to identify differentially expressed proteins. Whichever were differentially expressed ones, you'll take that protein and do analysis using mass spec. So this will only tell you like which were higher, which was lower, etc. Then mass spec is utilized to understand what exactly that protein is. So how exactly this technique is performed? So one of the technique is MALDI, which is matrix. So understand by you can understand it just by looking at the name itself, which tells that matrix assisted laser desorption or ionization technique. So there is a matrix here wherein you will add your sample to which is attached to the matrix. Now laser desorption or laser ionization. So desorption indicating it is coming out of the matrix. Initially it is on the matrix. As soon as you give laser to it, it will come like it will get desorption, and while desorptioning, it will also get ionized because you are giving a laser to it, and it will give certain charge to it. And now after desorption, it will uh, start going towards top wherein it will get separated. So there are demerits and advantages and disadvantages. Like sample preparation is easy here because you are directly adding the sample on the matrix, and more tolerant to salt than electron spray ionization. So this is laser assisted one. As I was talking, like there are different ionization techniques. So one can be ESI as well, wherein electron uh, spray ionization is done, and it is generally produces single charged ions. But the demerit is strong dependence on sample preparation method. So whichever your initial sample preparation you have done, accordingly those analysis, uh, those type of analysis will be seen in your results. Like those type of proteins will be seen in your result depending upon the way you have prepared your samples. So this is a soft ionization technique. It involves easy sample preparation. It is more tolerant to salt. It produces mainly singly charged ions, and it provides high throughput platform to establish what protein it is. So matrix है, then in that you have given laser to it, and it is now getting disrupted and as well as getting ionized. So it is running towards the analyzer. So top is a mass analyzer. So now here the separation happens depending upon mass to charge ratio itself, and time it takes for. So this so the smaller mass to charge ones will move faster here, whereas the larger ones will move slower, and that's how the separation takes place. And and time is also considered here. The time at which it is. So this is the path length basically from here till here. So this is the time taken for that specific ion to reach till the detector. There is a reflector as well, which will help in uh, moving the direction of the ions towards the detector. And as soon as it strikes the detector, now this detector will give us results in terms of spectrum. This is how you calculate the time basically. That this is what the time of flight equation is. So here the separation happens depending upon the mass to charge ratio. Generally, the lesser charge one will come out first, and whatever is the concentration, you will find that first. For example, even if this mass to charge is higher, but if the concentration is lower, you will find lesser concentration accordingly, like lesser peak size, and accordingly you can find. This is how the results are being shown. Like whatever is being hitting the detector, accordingly you will get the peaks continuously. So this results and depending upon mass to charge ratio measures mass to charge based on time taken by ions to fly in the analyzer and then strike the detector. The lighter ones travel faster and strike the detector before the heavier ones. This is the analyzer which we just talked about. There are different ways of it. Sometimes in order to increase the path length, people like in the detector itself, we like in the analyzer, there is reflection ion mirror as well, which takes longer time. For example, your protein is highly concentrated and it might have lot of proteins, or else it might have lot of peptides in that same mass or same sample, basically whatever you have done sample preparation for mass spec. So you need more time for it to get separated, or else everything will come and hit the detector, and you might get co-eluting peaks or something which came together and you are unable to separate them after. Sometimes that might happen. So having a reflection mirror helps, gives it more time to get separated, and the separation will be much more efficient. So this equation, if you want to know more details of it, this becomes mass of the ion. This is the charge of the ion cube. V is the accelerating potential, and L is the length of the flight tube. So this uh, will increase the time accordingly. So for this, this is the length which becomes greater here compared to the one which was closer. The detector was closer. Here the detector is here. It takes more time to hit the reflection ion mirror, and then to reach towards the detector, it takes more time. So it gets more time, the length is more, the time will be more. It is directly proportional. So how do we prepare the sample? So for example, this was the protein. You did the analysis and you found that this is a protein which was differentially expressed. For example, you took that, you have taken that gel, added it into the buffer, did in gel digestion, wherein you use trypsin generally. Then trypsin, how does trypsin breaks? Wherever there is lysine arginine residue at the C terminal, it will cut that specific peptide and make smaller peptides. So if this is the whole protein, it will make smaller peptides like this. Wherever arginine and lysine is present, it will make peptides like this. In trypsin digestion step, then you need to remove salts, buffer, detergents from the sample. Then we'll take the digested protein, add the matrix so that it gets attached with the uh, multiplate. So now you have added your sample to the matrix, and you will add those samples on the multiplate. This is how multiplate looks like, and you can analyze 196 samples at a go. This is the same thing, like this is the ionization, and this is where you keep the plate after loading your samples. Then the laser will come in contact with the sample, it will get dissolved and ionized, and it will move towards through the TOF, and it will hit the detector, and that's how you will analyze the samples. As soon as it hits the detector, you will get peaks accordingly. They have taken the gel, done the in-gel digestion, etc. So that is a process while doing in-gel dilation. We'll discuss that. So generally, wherever there is lysine arginine, that's what the cut happens. But when there is a proline after that, it is unable to cut. So trypsin has certain limitation. So trypsin cannot cut or break the protein if a proline is present after arginine and lysine, because that amino group is not free in order to interact and then cut the uh, uh, protein or like break the protein into peptides, smaller peptides. So in-gel digestion as a process, as I just mentioned. So initial step, you need to de-stain the gel because when you are cutting the gel, that gel is stained with combative stain or uh, whichever stain you are using. You need to de-stain that first. So by using this solution, you can de-stain that by incubating at 37 degrees for 10 minutes. Then you need to dehydrate also in order to remove all the water might be present. By using this solution, you can dehydrate by incubating accordingly. You have to reduce the proteins again with DTT and alkylate it again if there are because that's a separated protein. You need to repeat all of those process again. Then after doing reduction and alkylation, you do dehydration again. Once again by using acetonitrile, incubating, like following this specific protocol, and then you take that specific DTT reduced and alkylated sample for digestion, wherein you add 15 microliter of trypsin solution in the specific buffer and incubate for 4 hours until all the protein has been digested using trypsin. So what happens if your protein is like this, then you will break the disulfide bond using DTT. So now the bond is broken, and in order to make sure that they are not getting realigned or making the disulfide bond again, you will alkylate it again. Then you will take that specific alkylated protein to trypsin digestion. Then lysine and arginine is the one which will bands will be cut accordingly. Like if there is a proline, they won't be able to cut it, digest it basically. And after trypsin digestion, now you can find the concentration of your protein, like for of your peptides, etc., and then accordingly, depending upon your method, amount of specific concentration of protein is added to mass spec plate, like or else the MALDI plate after doing MALDI preparation, like adding it into the matrix, etc., and then loading onto the plate. So acetonitrile basically helps in dehydration of the gel and also helps in stain removal. Ammonium bicarbonate, where you are adding a buffer, provides an optimum pH for trypsin enzyme digestion. So these are the basically properties of how why you are using these specific chemicals. And overnight enzymatic digestion is also done instead of 4 hours. People might do overnight as well, which cleaves after arginine and lysine residue at C terminal. This is the location wherein trypsin acts. So general workflow is 2D and then MALDI-TOF. We just discussed MALDI here, and whether you use MALDI-TOF or LC-MS-MS, in-gel digestion is a crucial step wherein you actually because that is the one which will tell you what type of peptides are formed in your sample. This is a continuation of the same thing. You'll take the protein, do in-gel digestion, etc. Like in-gel trypsin digestion, and you'll do the mass spec. So this is just the visualization of the same thing. You'll use acetonitrile, ammonium carbonate, bicarbonate, etc. for de-staining it. Then you will add DTT, reduce it, and alkylate it using iodoacetamide. Then you'll do trypsin digestion. Everything is visualization again. Then this is how the fragments will be formed. Uh, then those peptides are added onto the plate. Like, so after that, you need to remove salts, etc. So this is a zip tip which helps in separation of salts and impurities, and only peptides remain here, whereas all other impurities are removed out by using zip tip. This is how it looks like. Sample cleanup are processed further using zip tip peptide pipette tips containing C18 or C4 media for enrichment of peptides. Now the salts and interfering agent, detergent, etc., are washed away using this type of uh, pipette. This is what we discussed basically. So matrix needs to be selected depending upon your type of protein. Then separation, matrix preparation, sample purification, sample desorption, matrix deposition, loading the sample, drying the target, and then doing the analysis. This is the whole protocol of matrix preparation basically. First, you need to select which matrix you will be using. You will prepare that. Then, whatever is the sample, you need to purify that, add it, mix it with the matrix, etc. Then, drying it onto the plate and then adding it onto the multi instrument for further analysis. So, these are the different matrix which are available depending upon your peptide might be present in your samples. If you have certain idea, then accordingly that type of matrix is used. For example, peptides less than 5000 Daltons or lipids and nucleic acid, you use alpha cyano matrix. If the proteins having higher than 5000 Daltons, then synaptic acid is used as a matrix. Then similarly, depending upon your need, if you are generally used for nucleic acid, if you are nucleotides, if you are dealing with nucleotides, small nucleotides and phosphorylation studies, then you use THAP and small molecules and peptides which are not ionized by other matrices. Generally use this so that it get ionized. This is how the plate looks like, and you're just adding a smaller amount of samples on each and every well which is there on the multi plate. So matrix preparation is done by mixing the matrix into a suitable solvent first, which we just done in the earlier slides, and vortex it first and few minutes to dissolve it properly, and then you load your samples. So you mix it with your samples and then load it like this. Then this is how the software looks like. So wherever you have loaded the sample, you'll just select that specific well and give information about that specific sample which you might know, and accordingly tell the instrument that target that specific sample and give fire as a command, and it will ionize that specific well, and it will get ionized and desorpted, and accordingly it will get separated, and you can do the analysis of that specific sample. This is how the spectrum looks like the M by Z, how separation might happen. So this is standard BSA protein which they have used. They have done the trypsin digestion of that specific protein, and they have done this analysis, and they found that this is how the spectrum or MS spectra was found after doing BSA proteomic analysis. So you can also do TOF, which will give you much more, what do we say, accurate results where if you are using TOF instead of using one TOF. So this is MS1 and MS2 basically. In terms of LCMS, MS, there is MS1 and MS2. Similarly here, this it is TOF and TOF, wherein it get coupled with two TOFs or hybrid Q-TOF you can say. So much higher sensitivity than TQ, which is triple chord, and single TOF. This uh, TOF is much more highly sensitive as I just talked about. So this is a software mascot peptide mass fingerprint which helps you all to do analysis of after separating this. So you can select each and every like the peak and accordingly do the analysis using this mascot peptide mass fingerprint. You can check samples, etc., compare them, and do all type of analysis what that peptide sequence is. If it is there, then how much it is there, compare with your control samples, etc. So generally higher the score, better is the hit. The hit should have at least two peptides. So for your you know the sequence, for example, you're trying to find your protein of interest. You know the protein of interest, right? Already. And if you see like the trypsin and lysine, digestion. So there are softwares available wherein you can just give the protein to that software. It will do the trypsin digestion. It will give you different peptides of those specific protein as well. Then you can accordingly check that protein like peptides and check whether those peptides are obtained in your mass spec analysis or not. So this is translational proteomics research. Like these are different applications of the same. So in terms of translation proteomics, it aims to identify novel diagnostic and prognostic markers that can be identified using this technique. It can help in focusing identification of novel therapeutic targets as well. If you have a certain long-term objective, personalized medication therapy in terms of healthcare. So all this can be achieved by using MALDI-TOF technique or LC-MS technique. So basically, there is a huge gap between bedside research, like whatever we are doing inside the lab, and bedside implication, indicating uh, the specific biomarker is actually went till the clinical trials and actually found that that is actually helpful for the treatment of patients. So there is a huge gap between that, often making it difficult to translate the findings into the actual applications. So how can we identify potential biomarkers by two different approaches? Either you can do backward approach or forward approach and do, which is also called as prospective and retrospective. So where what in terms of this is, so you have analysis retrospective wherein you will identify the target, the biomarker, and then that will be used for therapeutic responses to subgroups accordingly. But in terms of prospective, so there will be forward approach. In terms of forward approach, this is also called as prospective. The clinical trials, whatever results they are giving, so patient stratification will be the stratification indicating they will be divided like the healthy ones, the early stage ones, later stage ones, and try to find give a specific drug concentration and check whether if a patient is getting benefited out of it or not getting benefited. Accordingly, you can do the analysis of that. So there are different challenges associated with translational research. Translation indicating doing the work and actually getting translated to the uh, application of it. So in terms of translation, there is lack of standard protocols, cannot amplify proteins like PCR because in PCR, whatever concentration you are having, you need to do the analysis then and there. You cannot amplify and keep it like DNA, like PCR technique. There are presence of protein isoforms because of that, while doing the analysis, because of the isoform, you might be able to detect it, but actually it might not be present in that much high concentration, etc. So because of this, so there are different challenges associated with proteomics study. These are the few studies which are talked about. Then gave certain references as well, so you can deal with this in order to understand MALDI-TOF better. So that this were few of the topics which are discussed in this week 4 lectures. We'll take a break of like 5-10 minutes and deal with question and answers. Till then, if you are having any kind of doubts, so like in this lecture or any of the week 1 to 3 lectures, you can accordingly ask me. Till then, like let's take a break of 5 minutes or so, and then I'll start with question and answers. Okay. Okay. So can we start with the question and answer session? Yes, everyone. Yes. You all there? Okay. Can you all see the questions? Yes. So maybe we can start answering them. Okay. What is the advantage of 2D dyes over 2DE? Yes. Yeah. It's D. It's all of the above. So it helps in adding more. You can run more than one sample. There are less artifacts, obviously, because running one sample each will create more artifacts, and obviously gel to gel variation is reduced. Yeah, you can still see this. I think it's digestion. So select the correct order in in-gel digestion. So we read the like went through the process of in-gel digestion. So how is this? The process is destaining, rehydration, reduction, alkylation, dehydration, and digestion. Destaining, dehydration, reduction, alkylation, dehydration, and digestion. So you have prepared a protein sample for analysis using MALDI-TOF. Your sample have high abundance of phosphorylated proteins. Which of the following suitable matrix for your study you will be using? DHA, trihydroxyacetophenone. Yes. So all these have different benefits. We have gone through that in the lectures. Like if it is more than 5,000 Daltons, you accordingly use synapinic. Accordingly, they each have their own benefits. So psi dyes form covalent bonds with amino group for a particular amino acid. Which is that amino acid? Which of the following is the correct option? Yeah, lysine. Why lysine basically? Anybody elaborate on that? Like why lysine is preferred or why is like obviously Sidas will interact with lysine, but why was lysine? preferred generally for dyeing. Like instead of side dyes, people might have used any other type of dye as well, but they went with side dyes itself. So there were different benefits of that, and it. So currently, my question is like why lysine was selected. Yes, yes, everyone. I hope someone is typing too. No idea. We discussed about that, right? Yes, Veena, Himani, and Neha. If you cannot recall, because lysine is hydrophilic, yeah, like along with that, it is present in more majority of the proteins. At least one lysine will be present in almost all the proteins. That's the reason lysine was selected, along with obviously hydrophilic. It is outer side, it is easy to interact with the dye, etc. Which of the following enzyme can be used for proteolytic digestion? It's written as can be used, so what all can be used for digestion? Yeah. So everyone has their own way of digesting. We know trypsin, like pepsin and chymotrypsin has their own way of cutting, making peptides. So in it, MALDI-TOF analysis is mixed with matrix and spotted on multiplet. Match the following matrices. Okay, this is little complicated because you need to remember like what type of matrix is used for what. Yeah. So this was the first one, alpha cyano, which was used for. You just answered about THA, which is used for. Yes, the answer is A. 142231 and 4550 is third. After the control and treated protein samples are labeled with cyanide dyes, which of the following reagent is used to quench the unreacted dye? Because cyanide dyes interact with what? So obviously it will we need to add that specific reagent so that it gets interacted and the unreacted ones is also reacted now. Yeah. Which of the following parameter is used to create identical spot boundary across all the channels in Deitch data? Spot boundaries. Yes, everyone. So it's code detection, right? Because create an identical spot boundary across all channels indicating all three different dyes. So you need to find that make a identical spot boundary across all channels. So you need to have do code detection together. Then only you will be able to. So that happens during Dash data analysis. We'll upload all three images, and while doing that analysis, you need to code detect them in order to create an identical spot boundary. So zip tips used for sample enrichment, salt removal, sample containing digested peptides by associating column matrix because of which of the following interaction in the zip tip. Yes, I think you all can guess the answer as well. You don't have to be silent if you all are not getting what the answer might be. Still, you can guess the answers if you are not sure what might be the answer exactly. Yes, yes, Himani, Veena, and Neha. What is the interaction happening between when you are adding using a zip tape? The C18 gets interacted with the peptides. What is the interaction? It's hydrophobic interaction. Yeah. We have extracted protein samples with high salt contamination, so it is highly salt contaminated, but you do not have zip tips. Which of the following ionization technique would you prefer in order to remove the so that salts are also removed and your sample is also done properly? Analysis of sample. MALDI, that's what we studied, right? Which of the following is an advantage of dyes over gel-free approaches? So, although this gel-free approaches, I talked about it in maybe second week two lecture or something. Like there are different approaches for proteomics, but we are dealing with only gel-based one here. So, what are the advantage of this over gel-free ones? Gel-free like mass spec, you can still consider mass spec as gel-free if your basically your sample is not from the gel but just from the source, etc. Any blood sample or whatever, or purified by chromatography and then done the mass spec. So it can be done that way also. So you can consider that as a gel-free approach. So what is the advantage of dyes over gel-free? Yeah. So it basically detects modification of intact and undigested. It cannot say it as more sensitive because mass spec is more sensitive compared to dyes. So we want to do only dyes over gel-free approaches. So it detects modification of intact as well as undigested proteins. So don't go ahead after dyes you will go mass spec, then that then becomes gel-free approach. So it's still just the dye stage and then accordingly you need to give the answer for this. Which of the following criteria is used to select the matrix? Which of the following should be used to minimize any biasness in sample processing and dyes? Yeah, like if you apply a bit of logic, you will be able to answer this. We are providing PMF file generating using please analyze the following online mascot. So okay, this cannot be done now. What is the species of the sample? We need this specific software in order to uh, do this, but there is still you can answer this by referring to lecture 20 proteomics experiment data analysis. Then it would be easier for this. So currently we cannot answer this question though. This is the same thing. What is the approximate mass range? So basically, if you go through that mass code software or go through this lecture, it would be easier for you all to answer these questions because in that they have demonstrated how exactly mass code server works and how you can use this mass code software for analysis. The mass spectrometry can be used for what? Protein identification, characterization, quantification, or all of the above. You can do all of it. Which of the following solvent is used as a mobile phase in LC based experiments? So, like if you all go through the sample preparation step, you will be able to uh, answer this question. So, in generally, when you do liquid chromatography, acetonitrile is the one which is generally used as a mobile phase. Yeah. Which of the following is correct sequence of events in case of mass spec? Yes. In which of the following mass spec technique the sample introduced in solution form, it is eventually nebulized under an applied electrical potential? Okay, we have not done this, but still, if you can answer it. So this is a different type of ionization technique wherein you introduce your sample in a solution form and then using a nebulizer, it is applied an electric potential. So it's electron spray ionization. So in this technique, like your sample is in the solution form and it is in this like because of that spray under applied electric potential, they get ionized. The path of ions after deflection depends on. It's just a tricky question, but if you know the principle of how mass spec works, it's easy to answer. Yes. What is the path of ions after deflection? Depends on both charge and mass because mass spec runs on both mass to charge ratio and so until it reaches detector, it runs on the same principle. Yeah. So these were the few questions of week 4 lectures. So there's another course as well which runs on similar prospects. So you can just go through those few of those questions, not all of them. Like some might not be related what we have covered, but we can still go through few of them. So and conducted he used Psi3 dye to identify control samples, Psi5 below the representation of the image. So what does this show? Expression for protein A is unchanged. This is the protein. So you need to find what conclusion can be drawn by this study. This is how it looks. Protein A is upregulated in tumor sample. Protein A is downregulated in tumor sample. Protein A is differentially expressed in control and tumor samples. Which one? Yes. 3 and 4. It's 2 and 4, right? This is upregulated, right? Protein A is upregulated in tumor sample compared to this. It's not downregulated here. So this is A is upregulated than the other one. So this is the gel, how it looks like. Which of the following describe gel lanes 1 to 5 should be matched with the various types of run labeled samples? So you can read this question first. A collection of samples from two distinct conditions marked time point A and time point B, respectively, intended to use dice technique. Uh, so there are two time points A and B, labeling those time point A with Psi5 and those with point B with Psi3. The internal standard employed Psi2. SGS-based samples were collected both like pools of individual labeled samples and in individual samples. The gel was examined, the combined image. This is how it looks like. So when they visualize with one of the dye, etc. So which of the following describe how the gel 1 to 5 should be matched with various run labeled samples? They need to compare accordingly. So there are pooled samples as well. So if you see this fourth one, this is pooled sample of 1 and 2, right? So if you see compare the gel bands, if you see 3 and 5 are the same, whereas 1 and 2 is pooled together in 4, or else you can also say everything is pulled together in 4. You need to check the options accordingly and then answer, and you need to also know like which dye colors what. So the answer is 1 sci-fi, 2 sci-3, 3 is sci-fi, sci-3 and sci-2, all of them together. Fourth is sci-2 and fifth is Psi 5 and Psi 3. So you all can compare it accordingly. So time point, two time points, Psi 5 and Psi 3. So if this has everything, this is your internal standard. So that's the reason 4 is Psi 2, which is the pooled sample. This is Psi 5, this is Psi 3. Whereas as it is shown white in color, it has all the three together, Psi 5, Psi 3 and Psi 2, whereas fourth one is Psi 2 itself, fifth one is Psi 5 and Psi 3. Which of the following does this given image yellow and white band represent? So this yellow and white band represent what? So this is much more related to data analysis stuff, wherein the gel-based proteomics majorly deals with how those technique is performed. This is a different course though, but runs on a similar line. So I thought of if we can do these questions as well. These are little complicated questions, but in terms of analysis, how exactly those can be done, that's what that type of questions are present in this course. So proteins that are present in both samples from time points A and B are represented by these two colored bands. A protein present in the time point A sample are represented by both colored bands, but only the present time point B are yellow and the present in the sample time point A are in white bands. What do you think? So it's proteins that are present in both samples. Time point A and B are represented by this two colored bands. Now we don't know exactly because they haven't mentioned what that is possibly. Like it can be either A and B. So the now what we know in terms of the question is they are present in both samples from time point A and B and are present in two colored bands. This is again similar type of question. It will take time. Currently we are having 5 minutes only, so I'm just skipping this question. Which dye as depicted in lane 5 and 10 of gels? Oh, it's the same thing. Lane 5 and 10 are utilized to label the sample. So basically, in order to answer all these questions, you should know what Sci2, uh, like what color of Sci2 is in order to get visualized, or how Sci5 is visualized and how Sci3 is visualized. And you need to know bit of color chemistry as well. Like if you mix Sci5 and Sci3, how it will be visualized. If you mix Sci3 and Sci2, how it will be visualized. So accordingly, these colors are changing here, and that's what will help you to answer these questions. Okay, this we have not done in terms of degree and out yellow nodes, etc. So all these are not done in this course. Hmm. Okay, so those were the two. Okay, we can do this maybe, but also this has few other techniques. So we don't know exactly. Like we have not done in protein-based gel electrophoresis. Okay, I think these are different questions related to introduction to proteomics, but not gel-based one. Okay, so these were the few questions which were a part of this. What do we say? Specific week 4 lecture. So do you all have any kind of doubts for oh, I lost the contact. Internet. Yeah. Hello. Yeah, I just lost idea about the exam. So yeah, whatever is being uh, taught here, such type of like MCQs based questions will be asked probably. When I'm not sure exactly what type of questions are asked in the exam, but mm, like it depends upon the professor basically how he'll be designing the paper pattern, and maybe I can ask sir and email it to, or else I can add it in the announcement. Think how exactly the exam paper pattern will be, because that depends completely depends upon professor how he wants to design the questions. Yes, anything you all wanted to ask? And, so I think there will be one more. Is there a next week will we have class? So it depends. So do you all want a whole week one to week four quick revision, or else just question and answer like whatever we did again repeating those questions or adding few more questions if needed? Do you all want that class to be there, or like if you all need that, then I like we generally receive an email if we want to conduct any extra lecture for any specific course, and if we say yes, then only that specific lecture is scheduled, or else it is not scheduled. So generally this is week like 4 week course. So 4 weeks lectures are to be taken for sure, but the extra lecture depends upon us if we want to take it or not, depending upon your needs as well. Like the students who are enrolled into the class, if they need it, if TA is available, then accordingly TA make sure that he can take one more lecture. Yeah. If you all need that type of lecture, I can take that. So maybe we'll just go through different question and answers, or else if you want to get revised in brief about each and every technique, we can go through that as well. Depends. Okay, if you want it, I can. So I don't know when we will be receiving that email. Like if we receive that specific email, I'll generally say yes to it, and I'll give certain slot timing. So most probably I'll give same slot 7 to 9, but the day can change. It can be, so you will be accordingly receiving an email for that. Like if it is a extra lecture scheduled, then accordingly all can join that lecture. I'll be mostly keeping at the same time, like 7 to 9 on Monday itself. But if I have something else to do, then I might change the day, but timing would be most probably the same, 7 to 9. Yeah. Anything else you all wanted to ask me? Yes. I'll ask the professor, and I think there is an announcement uh, portal or something like wherein questions can be asked by you people. Uh, I'll try and reply in that specific thing because I don't have your email IDs obviously, but in the portal itself, I can ask the professor, like I can email professor and ask what can be the what type of questions can be asked in the lecture. I will try and make that like in the next week whenever we are having lecture. If I can get some information regarding that, I'll accordingly inform you. Okay. Anything else? Yes, Imani, do you have any doubts? I wanted to ask anything. Okay. I think you both were most probably there in most of the week lectures, like all four lectures. So you all can also give me a feedback accordingly if I need to improve on any of it so that my teaching skills get improved. What did you all understand whatever I was trying to say? Although it was just a revision kind of a stuff because sir has already uploaded those lectures and he has already explained it. We are just here to enhance that, like give a bit of more explanation if needed or if solving the doubts and etc. But still, like if you all liked it or not, etc., you can tell me accordingly. But thank you for attending the lecture for all 4 weeks. It was great to teach you all whatever I know about proteomics. Although I am in the learning phase of how exactly proteomics can be done, etc. But yeah, sure, whatever I know, I could just explain that. Okay. Anything else you all wanted to say? Okay, thank you. Thank you for attending. You can stop the lecture.